Welcome to your guide to Chapter 4 in Campbell's Biology on the topic of carbon and carbon compounds. The purpose of this chapter is to set you up and give you a good foundation for understanding the building blocks that are going to go into putting together these larger, very complex biomolecules that we're going to be talking about in the next chapter, and to understand why all of this really hinges on the properties of carbon and why carbon is such a special atom. So this will be broken up into three sort of chapters. The first will be talking about what are organic molecules and how did we come to have organic molecules on Earth. The second chapter is what makes carbon so special? Why is carbon such an important piece in this set of building blocks? And then finally, thinking a little bit about the things that we can stick onto carbon that's going to change the overall properties of the molecule in different ways. So these side chain groups that are going to make the molecule behave in very distinctive ways. So carbon really is the backbone of life. Living organisms consist mostly of carbon-based compounds. I mentioned at the end of the last web lecture that water and carbon are really the two things that it's very hard to even envision life occurring without these two substances. They are so fundamental to life as we know it. So carbon is unparalleled in its ability to form a huge diversity, almost limitless diversity of large, very complex molecules. And all of these macromolecules, these biological molecules that we're going to be talking about in the next two classes, are all composed of carbon compounds. They're all carbon-based molecules with different side groups attached to them that make them behave in their own special, distinct ways. So, the first chapter, Concept 4.1, the study of organic chemistry is really the study of carbon. So there can be different definitions of what an organic molecule is. Our book defines it as any carbon-containing molecule. Other definitions really focus on the role in living organisms, so any molecule that's only associated with living processes would be considered an organic molecule, but for our purposes we'll say it's anything with carbon. So, organic chemistry is the study of compounds that contain carbon, regardless of their origin. Organic compounds range from very simple molecules, think carbon dioxide, methane, CH4, to absolutely colossal ones. Remember that image of the humongous soluble molecule surrounded by tiny little water molecules. They can be really big on the molecular scale, right? We don't think of them as big from our usual frame of reference, but big for a molecule. Organic compounds are molecules that contain carbon bonded to other elements. So they're not graphite, which are just carbon. They're not diamonds, which are also just carbon. And the structure of carbon allows an almost limitless array of different molecular shapes. Different combinations of single and double bonds, as we'll see as we go through this, allow an almost infinite diversity of sizes and shapes of these organic compounds. And the formation of these carbon-carbon bonds was a very important event in what we call chemical evolution. So what is chemical evolution? Chemical evolution is an idea that attempts to explain the presence of these large organic molecules on the Earth, and then consequently leading up to the origin of life on Earth. So this is our leading hypothesis that we can actually begin to test the predictions of to explain the origin of life on Earth. So what is the idea? The idea is that inputs of energy led to the formation of increasingly complex carbon-containing molecules. So starting out with very simple molecules, remember that anytime you increase the complexity of those molecules, you're increasing the entropy, right? The amount of order. We know that that doesn't occur without an input of energy. So you need those inputs of energy in the form of light, in the form of heat, to be able to go from simpler molecules to more complex molecules. So eventually, these molecules would have become so complex just by coincidence of interacting with other molecules in the presence of an energy source. Eventually, one occurs that is a molecule that can replicate itself, that can make copies of itself. At that point, We can say that we've switched from a model of chemical evolution to a model of biological evolution. Once you start to have a replicating molecule, now it's subject to natural selection, and anything that allows this molecule to replicate itself more effectively, make more copies of itself, is going to be maintained in the population. Variations that cause that to happen less readily are going to decrease, and now we've got natural selection going. So then natural selection takes over. Eventually, a descendant of that original molecule became metabolically active, so it's able to sustain these reactions that allow it to replicate itself, and acquired a membrane, which, remember, is critical for maintaining homeostasis and also keeping those molecules that need to react together in close proximity to make those reactions more likely to occur. And at this point, we can say life had begun. Once you've got a membrane, metabolic processes, and replication, basically you have life. So now let's start to think about, is this even plausible? Is this something that can happen on Earth? And what is the evidence that this could happen? So we can never know for sure whether this did happen. This is not something that we can actually test, even with a time machine. But is there evidence that this could happen? Is this a viable hypothesis for the origin of organic molecules and the origin of life? So let's take a look at the evidence. So there are two general models for how this chemical evolution could have taken place. One of the earliest ones is this prebiotic soup model. So this posits that there were certain molecules that were synthesized from gases in the atmosphere or arrived via meteorites, which then condensed with rain and accumulated in the ocean. So that's the soup, right? This is basically an uncontained, everything just kind of floating around in these huge oceans. And that would result in this organic soup. So this soup that includes these very small, relatively simple organic molecules that eventually would collide together in the presence of an energy source to form even more complex, larger, more complex molecules. So this is the idea of that prebiotic soup model. And there were some early experiments to test some of the premises that this model was based on. Basically, can this happen? In the presence of a source of energy, can we get any kind of organic molecules from the substances that we know were present on Earth before life began? So a scientist named Stanley Miller, he was actually a graduate student at the time. He conducted these experiments along with his mentor. These experiments are typically called the Miller-Urey experiments. He wanted to answer a simple question. Is it possible? Can this even happen? Can complex organic compounds be synthesized from the simple molecules that were present in Earth's early atmosphere? So put another way, is it possible to recreate those very first steps going from simple inorganic molecules to the very simplest but still more complex organic molecules, creating those carbon-carbon bonds, for example? Can we do this in the laboratory? And so he set up a very clever apparatus to test this. So this apparatus consisted of an ocean. So this basically water that's being heated, because the temperature on Earth, remember, was pretty high at this point. And then an atmosphere in a separate chamber that includes the simple compounds, methane, ammonia, hydrogen, that we know were present in the Earth's atmosphere before life began. So as this boils, it's going to give off water vapor that's going to come into the atmosphere and react with these chemicals. It's equipped with an electrode that's going to provide a spark, so an energy source that will create these chemical reactions that decrease the amount of entropy, increase the amount of order, create more complex molecules from simpler ones. So then there's a condenser that brings water back out of this with these more complex molecules that we hope would be forming from this reaction. They're going to be dissolved in this water, and they're going to condense back out and flow back into the sea. We're going to take a little bit of it to sample to see what we made. The rest of it is going to go back in here to see if even more complex molecules are going to continue to be made from that. So what was the hypothesis of this experiment? If electrical energy is added to a mix of simple molecules, reactions will occur that produce more complex molecules, perhaps including some with carbon-carbon bonds. And here are the results. So from these samples that were taken for chemical analysis from the liquid water, some of them contain formaldehyde. So that's a slightly more complex organic molecule than what we started with. Hydrogen cyanide, another more complex molecule, and also glycine. So we do have that carbon-carbon bond coming up in a molecule that's quite a bit more complex than what we started with. And also this is an amino acid, so this is also one of the building blocks for the eventual creation of proteins. So the conclusion from this is that it can occur in a laboratory setup that simulates what we know about the environmental conditions of Earth before life came about, and it occurs rather readily if simple molecules with high free energy are exposed to a source of energy. So to take this just a step further, this is the idea. So you've got this ocean full of water, of course, and inorganic molecules such as bicarbonate that provide lots of raw materials for building more complicated organic molecules. You've got this atmosphere composed of carbon monoxide, carbon dioxide, hydrogen, ammonia, and then that gets exposed to light energy or maybe even electrical energy in the form of lightning, and that's going to form these larger, more complex molecules that are not going to be in the atmosphere. They're going to come back down into the ocean with the rain as the rain reacts with these molecules and brings them down into the ocean. These relatively simple organic molecules then are going to be exposed to heat in the oceans and just the normal collisions due to their kinetic energy to form these very complex organic molecules, eventually coming together to form something that can replicate itself, make copies of itself. Another model that scientists have come up with more recently is called the surface metabolism model. So one of the problems with this prebiotic soup model is that the soup is it's really big. And if these molecules are free to just float around uncontained in this humongous ocean, the likelihood that they're actually going to find each other and react in that particular way that's going to create more complicated molecules is it's a little bit unlikely. So this model tries to explain that or get around that problem by suggesting sort of a container, a more contained system that brings these molecules in close contact along with a source of energy. So let's look at how this works. So dissolved gases from the atmosphere that dissolve into the ocean come in contact with minerals that line the walls of deep sea vents. So these deep sea thermal vents that have a lot of heat, a lot of energy associated with them, they're going to form these more complex organic molecules in this context. To use light energy to synthesize these precursors, reactive molecules must have been localized and concentrated once they entered the ocean. This idea of just having them floating freely, able to disperse in the ocean, means that they're not going to be necessarily in the same place where they can react together. So the surface metabolism model proposes that these reactants were attracted to these mineral deposits in the deep sea vents. It also proposes that these minerals would act as catalysts to increase the rate of these reactions, hold these molecules closer together, and speed up the chemical reactions within the vents. And so preliminary research into this model shows that large carbon-based molecules can be formed under these conditions. For those of you who want to think about this more visually, here's the idea. So we've got simple molecules that have been dissolved in the ocean. Again, we're not talking about molecules in the atmosphere this time, but ones that have already been dissolved in the ocean. So dissolved carbon dioxide, protons, water. And this is going to be perfusing these deep sea vents, these little chambers within the deep sea vents. So if we look at one little section of this vent, it's got all of these little chambers and tunnels in it. And this is where these reactions are going to occur using the minerals as a catalyst. And when they're in these chambers, they're held close together and they can actually become concentrated in a way that makes these further interactions to create even larger, more complex molecules more likely, because they're all there in the same place. They're in a thermal vent, so there's a lot of thermal energy that's going to cause these molecules to move a lot and with great speed. That's going to make it a lot more likely that these molecules are going to encounter each other and run into each other in such a way that a reaction is going to occur. So stimulated by heat and concentration, the products of these more simple reactions are going to form even more complex molecules. So these are two possible models for how we might have gotten these larger, more complicated organic molecules present on Earth. So now let's shift gears a little bit and start to think about why carbon is such an excellent atom to form the backbone for these larger, more complex molecules. And the simple answer is that it makes 4 bonds. So with its 4 valence electrons, it makes the maximum number of bonds possible with the 4 electron orbitals that you find in these valence shells. And those of you who ever enjoyed playing with construction toys as a kid know that the connectors that are really going to allow you to build something really cool are the ones with that make the most connections, right? So this is going to make the most connections with other atoms of any of the elements commonly found in biological systems. So this is what's going to make these large, complex molecules possible. Just as an illustration, carbon will be able to make these long chain molecules by forming these carbon-carbon bonds with things attached to them, and this can be in pretty much any length. Carbon can also form rings by forming those 4 bonds, and then things can still be attached to those rings on either side. It's extremely versatile, and these rings can vary in size. Also, they can vary in shape, as we'll see. And in molecules with multiple carbons, each carbon bonded to 4 other atoms is going to have a tetrahedral shape. Just as we worked on in class when we modeled how those 4 electron orbitals are going to get as far away from each other as possible to create that tetrahedral shape. But we also saw in class that with a double bond, those 2 orbitals that are forming the double bond are going to kind of bend together and form a planar shape. So when 2 carbon atoms are joined by a double bond, the atoms joined to the carbons are going to be in the same plane as those carbons. It's going to form a flat shape. So to look at this a little bit more visually, so if we think of methane, it's a simple tetrahedron. Carbon single bonded to 4 hydrogens, it's going to form this simple tetrahedral shape. Ethane, even though there are 2 carbons, each carbon is going to form a tetrahedron around it. The other carbon will be just one of the sides of that tetrahedron, and you get this sort of 3-dimensional shape to the molecule. Ethene, or otherwise known as ethylene, on the other hand, is going to have a double bond between those carbons. Remember, those electron orbitals are kind of bent toward each other to share those 4 electrons between the 2 carbons. And so between this double bond and the 2 single bonds, those are all going to want to get as far away from each other as possible. With just 3 of these trying to be as far away as possible, it's going to form just a sort of a flat triangular shape between these 2. So the resulting molecule is going to be planar. So what does that mean? As you add more and more of these carbons, you can have single bonds or double bonds in various places. So you can have regions of this molecule that are flat versus regions that have this 3-dimensional structure and can be more bent. And that's going to lead to limitless diversity in the shapes that these carbon chains can take. You can get a lot of diversity arising from the variation in these carbon skeletons, the way these carbons are connected either to hydrogens or to each other. You can get variation in the length of these carbon chains, almost limitless. Numbers of carbons can be connected to each other in a chain. You can have variation in the position of a double bond. So be very careful when you're thinking about whether things are the same or different. If you're taking chemistry this semester, you'll work on this a lot. You need to think about whether you can rotate this molecule in some way that makes it exactly the same thing. For example, having a double bond here is not any different from having a double bond here at the other end of the chain. You could just flip it around and it's the same molecule. But if you have a double bond at the end versus a double bond in the middle, that's going to have a very different structure, right? This one is going to be flat on one end and have these tetrahedral shapes on the other end. This one's going to be flat in the middle, and then both ends will be a tetrahedron. So these are molecules with a different shape, despite having the same molecular formula. You still have 4 carbons and 8 hydrogens, right, in both of these, but it's going to have different properties. It can have different branching patterns. In this case, each carbon is connected to 2 other carbons, at least from the inside. This one of the interior carbons is connected to 3 other carbons. It's going to change the shape, change the properties of this. Remember, each of these is forming a little tetrahedron. It's going to look very different in this case than this case. The presence of rings. So being a ring rather than a chain is going to have a very different kind of shape and function. And so you can even introduce double bonds into these rings. In the case of benzene, you have alternating single and double bonds. So what you'll notice is every single carbon in this ring is participating in one double bond. That's going to make this whole ring flat, as opposed to this 6-carbon ring that's going to be sort of undulating because of the tetrahedral shape of all of its bonds. Another way you can get a lot of diversity in these carbon chains is the idea of isomers. If you're in chemistry this semester, you'll be working on isomers a lot. Draw all the different possible isomers of a molecule with this chemical formula. It's actually kind of fun figuring out all the different isomers, but we just want the general idea that you can arrange these molecules that have the same molecular formula but give them very different structures and different properties. So we can have structural isomers that have different connections between the carbons. So these, all the carbons are in a straight line. All the interior ones make bonds with just 2 other carbons, as opposed to a structure where you have a branch in the carbon chain. So one of these interior carbons is connected to 3 other carbon atoms. Cis-trans isomers have the same covalent bonds but differ in their spatial arrangements. In the case here of cis-trans isomers, you've got your functional groups on the same side of this planar molecule. Remember, with the double bond between the carbons, you get a planar molecule. In this one, you've got them on opposite sides of the carbon double bond. There's no way to rotate this around to make this look the same. These are different isomers. They're going to behave differently, particularly in the interaction between these 2 side chains, right? They're going to be close together in this case. They're going to be far away from each other in this case. They're not going to interact in the same way. The enantiomers are a little bit more difficult to see. So these are isomers that are mirror images of each other. And the best way to visualize this is, for both of these, put this carboxyl group on the top, and then start with your hydrogen and start going around clockwise and see which other functional groups you come to as you go around clockwise. So if you start here with hydrogen, the first one you come to is the amino group, followed by the methyl group. Again, putting the carboxyl group on top, starting with the hydrogen and going clockwise. This time, first you're going to come to the methyl group and then the amino group. So these are, we think of this as left-hand and right-hand mirror images of each other. They're going to have slightly different properties. And your book goes through a lot of examples for how these different isomers are very important in the pharmaceutical industry, and that is a great illustration of how, even though they have the same molecular formula, all the same atoms involved, they're going to have different properties and do different things in the biological systems. So next, let's start to think about the kind of diversity that can be generated as different groups of atoms are attached to these highly varying carbon skeletons. So a few chemical groups are the key to most molecular function in biology. So the distinctive properties of organic molecules depend on the carbon skeleton and also the chemical groups that are attached to it. So a small number of characteristic groups can replace the hydrogens that are attached to the carbons in those organic skeletons and really change the behavior of those molecules. For example, estradiol and testosterone are both steroids with a common carbon skeleton in the form of 4 fused rings. So we see our carbon rings here. In this shorthand, each of these corners represents a carbon in the carbon skeleton. If you see a line here, that means there's a double bond, so that tells you something about the shape of that molecule in that area. We see the same sets of, in this case, 3 6-carbon rings attached to 1 5-carbon ring in both of these. But they have different functional groups attached to them. So these sex hormones differ only in the chemical groups attached to the rings of the carbon skeleton. So in this case, an OH, and in this case, an oxygen. So we can see right off the bat that that's going to change the shape of this carbon ring because we have fewer double bonds in the carbon ring. All of these places without double bonds are going to have a 3-dimensional shape to them. Wherever you have the double bonds, you're going to have a planar shape, and we already have a slight difference in the shape of these molecules that's going to change the behavior of these 2 sex hormones. But to start to think about these side groups, let's start to think about when there aren't any of these side groups and we only have hydrogens attached to the carbons. So hydrocarbons are organic molecules consisting only of carbon and hydrogen. So this is kind of the simplest case. We see this at least as parts of many organic molecules, such as fats, are going to have long hydrocarbon chains. So if we think about this is a fat cell with a nucleus here and little vesicles containing these fat molecules that are going to have these long hydrocarbon chains. So being composed of only carbon and hydrogen, these are of course nonpolar, and they also contain a huge amount of energy. So hydrocarbons can undergo reactions that release a large amount of energy, so there's a lot of stored potential chemical energy in these bonds. So that's why fat is so useful for energy storage. Hydrocarbons also make up most of the carbon-based combustion fuels that we use, the gas in our car, the cooking gas on our stove. These are all based on combustion of hydrocarbons and the huge amount of energy they store in them. So as we move away from these simple hydrocarbons, we're going to start to replace these hydrogens with different functional groups. So these are going to be important compounds containing different combinations of hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And they're going to come in just a small number of different functional groups that are going to be important to the biology we're going to be learning. As always, a heads up, when you're going to have to memorize something, you're going to need to memorize these functional groups, you're going to need to know their chemical formula, and you're going to need to know their general properties in the molecule. So make sure that you are doing whatever you need to do to make sure that you have these committed to memory. What's an amino group, for example? So here's just a list of some basic properties of these different functional groups. Amino groups attract a proton and they are going to act as bases. Carboxyl groups are going to lose a proton and act as acids. Carbonyl groups have sites on them that are going to link different molecules together into more complex compounds. So that's the role they're going to tend to play in these large complex molecules. Hydroxyl groups act as weak acids, so they're going to be proton donors. Phosphate groups have 2 negative charges, so they're going to have a high affinity for positively charged particles. Sulfhydryl groups link together to form disulfide bonds. We'll see a lot more about this, particularly when we get to nucleic acids. And methyl groups actually are just hydrocarbons, but we are thinking about them as a functional group because they're a group that includes carbon with 3 hydrogens attached to it. And we think of this as a group that's attached to a different carbon chain rather than that carbon being part of the carbon chain. And the reason we do is because this attachment of a methyl group, or what we call methylation, is going to have a big effect on the molecular shape, and it's becoming increasingly understood to be an important factor affecting gene expression. The attachment of this methyl group can change the way genes are expressed. And so we're going to think about them as part of this group of functional groups, even though it is just carbon and hydrogen. So here are some of these and their chemical formula and their chemical structure. So a hydroxyl group is basically just OH. And in a compound, these are called alcohols. So an example is ethanol, which is a 2-carbon chain with one of the hydrogens replaced by a hydroxyl group. And again, remember that this is going to act as a weak acid. So when you have an OH group, it's going to behave like a weak acid. Carbonyl group is just an oxygen double bonded. So it's going to replace 2 of those hydrogens and create a planar region. Don't worry about getting too complicated with understanding the exact shapes that are going to be formed by these really complex molecules, but what I do want you to understand is introducing a double bond is going to have an effect, changing that shape from a 3-dimensional shape to a planar shape. So that's kind of the level I want you to understand, that this is going to be different than something with 3 single bonds. It's going to have a different shape. So this carbonyl group in a compound, this is going to be called a ketone or an aldehyde. And some examples are acetone and propanol. You can see in acetone we've got that oxygen double bonded in the middle of the carbon chain and propanol at the end of the carbon chain. So these are both carbonyl groups. The group is just an oxygen double bonded to that carbon chain. And again, this is going to provide sort of an attachment site where that oxygen can bond to something else and link things together into more complex compounds. A carboxyl group is going to have a double bonded oxygen, and then the one hydrogen that would have been bonded to the carbon if this had been a carbonyl group is going to be replaced by an OH. So we call this COOH, the 2 oxygens and the 1 hydrogen, and this is going to form, again, a planar region in the molecule. And in a compound, we call this a carboxylic acid or an organic acid. And obviously, this is going to act as an acid. So acetic acid is an example. It's going to lose that hydrogen. Remember, this is a highly polar area with the oxygen pulling the electrons away from the hydrogen, which is going to tend to dissociate. Amino group is an NH2 group attached to the carbon chain. So just single bonds, one with the carbon, skeleton, one with each of the 2 hydrogens. We call this an amine. When it's in a compound, one example is glycine, and these amine groups are going to tend to attract protons to this electronegative nitrogen to form an ionic region, NH3+, attached to the carbon that's going to act as a base and accept that proton. So one thing I want you to notice in these side groups so far is that all of them introduce polar covalent bonds into these hydrocarbon chains, which were initially nonpolar. So oxygen, remember, is the most highly electronegative. It's going to create a very polar bond both with the carbon and with the hydrogen. So we've got a polar region anytime we've got a hydroxyl group or any of these groups, wherever they occur, because of those differences in electronegativity between oxygen and nitrogen, and also phosphorus and sulfur, between them and hydrogen and carbon, which are not very electronegative, anytime you have one of these side groups somewhere on your molecule, you have introduced a polar region to that molecule, and that's going to change the behavior of that molecule as well. Looking at the other 3 groups that we need to know about, one is a sulfhydryl group, and that's just a sulfur and a hydrogen attached to that carbon skeleton. Remember, sulfur has a valence of 2. In a compound, we call this a thiol, and an example is cysteine. And these sulfurs are going to tend to bond with other sulfhydryl groups to form a bond between the 2 sulfurs, and we call that a disulfide bond. Again, we'll see this in more detail when we get to nucleic acids, because those are going to be very important in the structure of nucleic acids. Next, we have phosphate groups, and this is an oxygen connected to the carbon, and then Attached to that oxygen, there's going to be a phosphorus with 3 other oxygens. And again, these 2 oxygens are going to have a negative charge, so it's going to have an overall charge of -2. So we call this an organic phosphate when it's attached to a carbon chain. And an example here is glycerol phosphate, where we can see this phosphate group attached to the carbon chain. So again, those 2 negative charges are going to be really important in these phosphate groups. And then finally, the simple methyl group. Remember I said that that was just a carbon with 3 hydrogens attached. And we call this a methylated compound. Remember I talked about methylation. And here's an example of a carbon ring with some nitrogens attached in the ring and a methyl group attached to one of those carbons in the ring, 5-methylcytosine. And again, these methyl groups are going to be really important when we start to think about gene expression. They're going to affect how genes are expressed. Before we leave the idea of functional groups, I want to bring your attention to another behavior of these phosphate groups in forming a molecule called adenosine triphosphate, or ATP. So ATP is super important as the direct source of energy that cells actually use to make things happen, to do the work of the cell. So all of the chemical energy that's taken in has to be converted to ATP before the cell can actually use it. So ATP consists of an organic molecule called adenosine. We'll learn a lot more about that as we go along, attached to a string of 3 phosphate groups. And so as one of these phosphate groups is removed, energy is released that can be used by the cell. So ATP stores the potential to react with water, so this is going to happen in the presence of water. And again, one of those phosphate groups is going to be removed, forming ADP, adenosine diphosphate, plus an inorganic phosphate, releasing a bunch of energy in the process. So let's just wrap up with the sort of 3 major points from the 3 different chapters of this web lecture. First of all, chemical evolution is a very useful framework to start to think about how we could have initially had the presence of these more complex organic molecules, leading to even more complex molecules until eventually one formed that was capable of replicating itself. After that point, biological evolution by natural selection would take over and favor anything that tends to increase that replication rate of the molecule. We saw that the versatility of carbon makes possible the huge diversity of organic molecules, almost mind-boggling diversity of different organic molecules that make up all of the diversity of life on Earth. And we also saw that variation at the molecular level lies at the foundation of all biological diversity. So variation in the structure of these carbon chains and also in the different functional groups that can be attached to these carbon chains is going to underlie all biological diversity. So from here, we're going to start to look at the actual biological molecules that make up organisms and see how this variation in structure, how the differences in these side groups and their behavior cause these different biomolecules to behave in the particular ways and belong to the certain categories that they belong to.